Good afternoon, everyone, and welcome to Genome Webinars. I'm Ben Butkus, Editorial Director at Genomeweb, and I'll be your moderator today. The title of today's webinar is Spatial Profiling to Assess a Novel Gene Therapy in a Mouse Model of a Rare Genetic Disorder, and is sponsored by Advanced Cell Diagnostics. Our speakers today will be Maggie Wright, a scientist at Ultragenyx Pharmaceutical, and Anushka Dikshit, application scientist at Advanced Cell Diagnostics. Attendees may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. If you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. With that, I'll turn it over to Maggie Wright of Ultragenyx Pharmaceutical.
Hi, Ben. Thank you very much for that introduction, and thank you to the organizers and to ACD Bio for sponsoring this webinar. I'm really excited to talk with you all today about some of the work that we've been doing to test one of our novel gene therapy drug candidates in a mouse model of a neurological disease called CDKL5 deficiency disorder. So first, I wanted to start out with my disclosures. I'm an employee of Ultragenyx Pharmaceutical, and all of the studies that I will outline and review in this talk were sponsored and funded by that company. So to give you a little bit of background on what CDKL5 deficiency disorder is, it's an X-linked neurodevelopmental disorder, and this is caused by loss of function mutations in a gene called CDKL5. This gene codes for a protein product called cyclin-dependent kinase-like 5 gene, and this protein is expressed ubiquitously throughout the body, but one of its most important locations of expression and of function is within neurons. And if I can call your attention to the schematics that are on the right side of the screen, the little kind of hairy orange guy is a diagram of a typical neuron. And neurons are composed of three basic functional parts. They have the cell body, which has the nucleus and all of your typical organelles. And then they also have two different types of neurites, or extensions. They have dendrites, which are responsible for receiving information from all of the neurons around it. And then they have an axon, which is important and required for sending out the information from that neuron to all of the neurons that it will then communicate with. So dendrites are the particular location that CDKL5 is really important for developing and maintaining for individual neurons. And each neuron actually has the capability of contacting hundreds of thousands of other neurons. So the ability for this cell to synapse with all of those different neurons is really important for its ability to communicate and function as a neuron. So to create these synaptic contacts between other neurons and this neuron of interest that we're looking at right here, these dendrites have dendritic spines that, oh, it looks like attendees are not seeing the slides. I am moving forward here. Let me, all right, can you see the CDKL5 deficiency disorder slide?
Maggie, we can see that slide now, the CDKL5 slide.
Okay, sorry about that. I'll keep going. So yes, dendritic spines. Those are the important synaptic contact location for neurons when they're communicating with other neurons. So CDKL5 is important as a kinase, which means that it phosphorylates other proteins. And it was when it phosphorylates those substrates, it tells them to do certain functions. And what CDKL5 is doing here is important for phosphorylating substrates such as microtubule binding proteins that then are responsible for forming the dendritic spines and maturing them. So in the absence of functional CDKL5 protein, these dendritic spines have this like immature small shape, and they don't have the proper complement of receptors that are necessary for them to communicate with other neurons. So in the absence of this protein, these cells form really immature dendritic spines that prevent them from properly communicating. So this results in a really complex neurodevelopmental disorder known as CDKL5 deficiency disorder. And up until recently, this disease was actually grouped in either a generalized autism spectrum disorder phenotype or as an atypical form of Rett syndrome. But there are a few things that are very specific to patients who lack functional CDKL5 protein that makes this a unique disease that we've then been generating a therapy specifically for. So the big thing that arises in these patients very early on is that they have a really incredible seizure burden that starts before they're even 3 months of age. And these seizures, unfortunately, are not responsive to typical anti-epileptic drugs. They're really quite severe, and they persist for the lifetime of the patient. This is one of the things that has actually been helpful to getting these patients diagnosed, though. CDKL5 deficiency disorder, or CDD as I'll continue to refer to it, is now on diagnostic panels so that when kids start to have these seizures very early on, they're now identified much earlier. And this is really helpful so that we can get in with some earlier interventions as clinicians. So in addition to the seizures, these kids also have a generalized global developmental delay. All of the developmental milestones that parents and caregivers look for when they're watching their children grow, many of them just are never met in these patients. They lack many gross and fine motor skills. Very few of them can crawl, and even fewer of them are able to walk independently by the age of 5 years old. That typical pincher grip that you're used to seeing kids use when they're sitting in their high chair and grabbing at their Cheerios, this is something that these kids they can't coordinate to do that type of movement. In addition to these motor developmental delays, these kids also have very impaired communication ability, and they have a generalized intellectual disability. In addition to that, there's also some things that are specific to this disease, such as vision problems, feeding difficulties, breathing difficulties, and sleeping disturbances. There's been anecdotal reports from patients and their parents that say that they'll go upwards of 24 to 48 hours without sleeping a wink, which you can imagine is an incredible burden on these caregivers, and something that's just, it's not a good pattern for these kids to get into. So it's expected that this disease has an incidence of about 1 in every 40,000 live births. And as we're learning more about it, we're also finding more patients. And about 2 patients are being newly diagnosed every week since we now have this on the seizure panels. So here is the therapeutic approach that we are taking to go after the root cause of this disease. What we're doing is we're testing a gene therapy. We're using an AAV9 viral vector that encodes or encapsulates a human form of the CDKL5 gene that's being expressed by a synapsin promoter. And this is important because the synapsin promoter will cause this gene to only be expressed within neurons, which is the cell type that we think CDKL5 is really required for. So the route of administration that we're going to be studying here, or we're going to be analyzing, is by administering this virus into the cerebroventricular space where the CSF fluid is. And we think that by going into this space, it's going to give us the best opportunity to hit as many neurons as possible. Now, when we're looking at the mouse model of the disease, we're going to be administering the virus into the lateral ventricle, which you can see on the left-hand panel of the screen. This gives us the biggest amount of space that we can put the most amount of vector into. However, when we think about going into patients, we're not going to be going into the lateral ventricle. There's too much brain matter to have to get through, and that would be a very onerous procedure. Instead, what many groups are doing is they're going into the intrathecal space, either by a lumbar puncture or up just on the backside of the brain in a space called the cisterna magna. So once this vector is administered into the CSF space, it then travels through until it binds to a receptor on the outside of a cell, such as a neuron. This virus is then encapsulated and brought into the cell, where it's then transported into the nucleus. The viral particles are shed, and then the actual CDKL5 gene is read. It's made into mRNA, which is then produced into protein. And this protein product is what we're really excited to be able to replace to these cells, as that is the actual root cause of this disorder, the lack of functional CDKL5 protein. So the mouse model that we are using to test this particular gene therapy in is a model of CDKL5 deficiency disorder where exon 6 specifically has been knocked out in these mice. And there's two different genetic models that we'll be looking at. There's hemizygous males that have no CDKL5 expression, and they have a bit more of a severe phenotype. Females, however, are heterozygous, and you'll remember that I mentioned this was an X-linked neurodevelopmental disorder, and that's because when this gene is absent on one of the X chromosomes, half of the cells in these heterozygous females will have normal levels of CDKL5 protein, while half of them will have no functional CDKL5 protein. This is due to X-linked inactivation, and the majority of CDD patients are in fact females that have this kind of mosaic phenotype where half of the cells are normal and half of the cells are lacking the protein. So both of these models give us different abilities to look at different things. The males are helpful because they have a more severe phenotype, and that really allows us to see if we can move the bar on with application of this treatment. However, the females are a little bit genetically more like the patients that we're going to be aiming to treat. So both of them have value for us when we're thinking about testing this therapeutic. Now, before I get into some specifics of the data, I really wanted to call out the intention with which we designed our dosing paradigm in this mouse model. So many groups, when they're testing gene therapies, what they do is they go in very early. They go in at like the first day of age with these mice, with their gene therapy. They administer it, and they get huge levels of robust transduction. The virus goes everywhere, and that's because the blood-brain barrier isn't totally formed. The tissue is smaller. It's just a much better transduction profile. However, when we're thinking about translating all of the studies we're going to do in a mouse to patients, we really wanted to be intentional in designing these studies to be as easy to translate to that patient phenotype as is possible. So by going in at 3 to 5 weeks of age, we know we're not going to hit every cell. That's just not what the technology is able to do right now. And we know that these mice already have some symptoms. But these are the two pieces of the puzzle that we know are going to be the case when we're looking at translating this to patients. We know that by the time we get to these patients, they're already going to have symptoms. And we know that with the limitations of this technology, we're not going to be able to hit every cell. But by testing these same limitations in a mouse model, if we're able to move that bar at all on the phenotype of this mouse, it would give us confidence that we're able to do so in a patient as well. So before we went into our crux studies looking at behavioral phenotypes, the first thing we did was, using this mouse model, we wanted to see if we were able to express functional CDKL5 protein. So to do this, we did an intraparenchymal injection of our vector straight into the brain matter of these mice. And by doing this, we know we're going to be expressing a lot of protein. And by looking at those high levels of CDKL5 protein, we'll then be able to look downstream and see whether or not it's functioning appropriately. And I also want to call out that all of the studies I will be describing here were done in collaboration with Dr. Rodney Samako's lab at the Baylor College of Medicine. They've been an incredible lab to collaborate with, and this has really been one of those instances where industry and academia can really work together and leverage our abilities and our strengths and our weaknesses to create a really complete body of work that we're excited to hopefully have published in the near future. So this is a busy slide, and I want to walk you all through here. On the left side, there's a cartoon schematic of a sagittal section of a mouse brain. So when I say sagittal section, it's right down the midline, as with the front of the brain on the left side of the screen and the back of the brain on the right side of the screen. And if you look at the fluorescent image on the right of this, this is an RNASCOPE in situ hybridization for human CDKL5 mRNA. And I want to say that this is a technology produced by ACD Bio, which Anushka will talk about after after this particular part of the webinar, this really gave us some power that we weren't able to do without this particular technique. So what we wanted to do is we wanted to see where this virus was going and where it was actually transducing cells, creating mRNA, and creating protein. However, there's no good antibody that's currently available that allows us to look for protein levels of CDKL5. So with RNASCOPE, what we're able to do is we're able to look at the next best thing, the mRNA. And what you can see with all of that red signal is that we have a robust amount of human CDKL5 mRNA in the cortex, and then right underneath that, that big red blob, that's in the striatum, which is right around where we actually did the actual injection of the virus. So this was really exciting to us because we're able to see robust levels of this mRNA being expressed off of our viral product. So now that we know that we're getting transduction and expression, what we wanted to do is we wanted to look at different areas of the brain and see if we're able to move the bar on any functional downstream targets of CDKL5. So the one that we chose was EB2, which is a microtubule binding protein that's recently been identified as a direct substrate of CDKL5. So if I can call your attention down to the bottom middle of the screen, this is a Western blot looking for protein of CDKL5 and of phospho-EB2 from wild-type mice that are treated with PBS, from CDD mice that are treated with our AAV9 vector, and then lastly with CDD mice that are treated with PBS. If you look at the first set of bands, you can see that CDKL5, if you look all the way at the right, that band is absent in the CDD mouse that's treated with PBS. However, if you look towards the middle, CDD mice that are treated with our AAV9 vector have robust levels of the protein, making us confident again that this our viral vector is actually producing a CDKL5 protein. So to test its ability to phosphorylate its downstream targets, we then looked at levels of phosphorylated EB2. And again, if you look all the way on the right, you can see that in that middle row, the levels of phosphorylated EB2 in a PBS-treated CDD mouse are really low, almost undetectable. However, if you look at the CDD mice that were treated with our AAV9 vector, you can see that those bands are almost identical in level to that of the wild-type mice treated with PBS. And this is quantified and normalized to levels of total EB2 in the graph on the right. So this was really exciting to us because we're seeing where this protein's being expressed, we're seeing how much it's being expressed, and that it's actually functional in being able to phosphorylate this EB2 protein. So the next big study that we did was an injection of this virus into the lateral ventricle of male and female mice, again around 3 to 5 weeks of age. And then 2 months later, we did a behavioral panel on them to see if we were able to correct in any way the phenotype of these mice. And then we also again looked at the biodistribution of this virus, since this is a different route of administration. We want to see by going into that CSF space where exactly the virus is going. So here is, again, an RNASCOPE image showing where the human CDKL5 mRNA is present within a sagittal section of an AAV9-treated CDD mouse. So what I hope you can appreciate by looking at this image versus the one I showed you on the prior slide is that we have human CDKL5 mRNA throughout the brain, and we've got a lot of it in the hippocampus in particular, which we know is an area that we're really going to want to target when we eventually think about translating this to patients. So once we felt confident that we were seeing robust mRNA throughout the brain, we then went ahead and did Western blotting at different portions of the brain to look at protein levels. So we looked at CDKL5 in the frontal cortex, in the hippocampus, and in the brainstem. And what we saw is that in all three of these regions, we were able to increase the amount of CDKL5 protein that's present here. And we were able to quantify this as being somewhere between 20 and 35%, which we were really excited about. We know that, again, we're not going to hit 100% of CDKL5 protein by doing this particular dosing paradigm. But knowing that we can move it even by this much gave us confidence moving forward that we were going to be able to hopefully have some translatable effect when we look at their phenotype. So before we took these mice down, what we did was a behavioral panel to assay the phenotype of these mice. Different labs have looked at different tests, and they've seen a phenotype of this CDD mouse model that's different from their wild type siblings. So what we did is we ran these tests, and we focused on the ones where the wild type mice and the CDD mice that were treated with PBS were statistically different from each other. And this tells us that there's a baseline phenotype. So what we did then is we looked at the CDD mice that were treated with vector. And these are both male and female. We've grouped them together for these particular analyses. And we wanted to see how they changed relative to these two treatment groups. So we binned them by 3 categories. They either showed no improvement, partial improvement, or their behavior was normalized. What no improvement looked like is that the CDD mice treated with vector performed the same as CDD mice treated with PBS. What partial improvement looked like was that we were able to move the bar just a little bit down. So these CDD mice treated with vector were no different than wild-type mice or CDD mice. However, we saw that they were normalized if we were able to move the bar all the way down, and CDD mice treated with vector were no different than wild-type mice. This is ultimately our goal, but by binning these types of analyses in these 3 ways, it allowed us to see even if we were able to change the phenotype slightly, even if it wasn't able to get all the way to wild-type levels. So 2 of the motor functional tests that we looked at were the parallel rod floor plate and the open field test. In the parallel rod floor plate, we put mice into the structure on the left where these rods go across the bottom of the floor. And every time that the mouse walks over it and their feet fall through the rods, it's automatically detected and it's quantified. What we saw was that CDD mice slip a little bit more frequently than wild-type mice through these through the rods, which indicates that they have some motor coordination deficits. We also looked at an open field test where these mice are placed into this box here that you can see, and then they're allowed to freely navigate for about 30 minutes. And then the distance that they travel and the number of times that they're moving and such is quantified automatically. And this is consistent with what other groups have seen in that the CDD mice have a hyperactivity phenotype. They walk around this open field much more and travel much further than their wild-type litter mates. Now, when we quantified the activity levels and the footfalls of the CDD mice that were treated with AAV9, what we saw was that we were able to move the bar a little bit, but not all the way down to the wild type. And this is what we saw as partial improvement, as I outlined on the prior slide. And this was exciting because we were able to see in 2 different motor functional assays that we were able to fix the phenotype a little bit, even though it's not all the way down to wild type levels. So another assay that we looked at was the cued fear memory assay. And what happens in this particular test is that mice are put into a training environment where they learn to associate a tone with a foot shock. And then 24 hours later, they're put into a different environment, but given the same tone. And what this test does is it assays the ability of these mice to remember and recall that that tone is associated with a foot shock. If these mice are able to remember that, if they have that ability to recall what they learned the prior day, they freeze. And that's what we do, is we quantified the amount of time or the percentage of time that they spent freezing. So wild-type mice, when they hear this cue, they remember it and they freeze for about 60% of the time that they're in this box. CDD mice that are treated with PBS aren't able to perform as well. They're not as able to remember that this tone is associated with a foot shock. However, when we tested CDD mice that were treated with our virus, what we saw is they performed identically to that of wild-type mice. Their memory was performing much better than that of CDD mice that were treated with PBS. And I've highlighted here some RNASCOPE images that are looking at 2 of the structures in the brain that we know are particularly important for this type of memory, the hippocampus and the amygdala. So this was encouraging to us, knowing that we were able to target those regions in a way that was relevant to the phenotype of these mice. So in summary here, I wanted to outline that our therapeutic approach of gene delivery to the CSF of early symptomatic juvenile CDD mouse model, we were able to show sustained human CDKL5 mRNA presence in neurons throughout the brain up to 4 months post-injection. And I can say that in some of our subsequent studies, we've also seen it for much longer. And this is important when thinking about the durability and the length of a gene therapy in bringing it to the clinic. Moreover, we were also able to show sustained levels of functional human CDKL5 protein within neurons and within multiple disease-relevant brain regions here. And in addition to this, we were also able to show that there were multiple behavioral domains that we were able to move the bar and show that this level of CDKL5 protein replacement was actually functionally relevant and beneficial for this mouse model. So I want to highlight that even though we're not restoring all of the behavioral deficits, this is really important data for us to think about moving forward, showing that even a small amount of CDKL5 protein replaced into this mouse model is helpful. And in thinking about the limitations that we have with our current gene therapy technologies, even knowing that we can replace just a little bit of protein potentially back into these patients could be really helpful and life-changing for the quality of life for CDD patients and for their families. So this is a really encouraging thought for us moving forward for this particular disease and for this particular approach. I want to end by acknowledging all of the people that did this work. A number of us at Ultragenyx Pharmaceutical, in addition to Rodney Samako and his lab at Baylor College of Medicine. Studies like this take a village, as I'm sure you're all aware, and I've been really excited to work with some very talented and excited people about this project. So yeah, that's it.
Thank you, Maggie. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We'll now turn the webinar over to Anushka Dikshit, Application Scientist at Advanced Cell Diagnostics. Please go ahead, Anushka.
Thank you, Ben. Can you all hear me okay?
We can hear you, Anushka.
Great. So after that brilliant presentation by Maggie, I'm going to take some time to talk to you about our RNASCOPE in situ hybridization platform and help you understand how you can utilize this technology to expand the spatial biology research in cell and gene therapies. So to start with, let me talk to you about what our platform entails. So for those of you who are not familiar with the RNASCOPE technology, it's an in situ hybridization platform that was specifically designed to overcome some of the traditional limitations of an ISH assay, which include high background, non-specific signal, and so forth, while still giving you that precise detection of your target gene. So when we developed the RNASCOPE in situ hybridization assay, the goal was to provide an ideal spatial biology solution to identify RNA biomarkers that would helpfully advance therapeutic strategies in a number of pathologies. So the three major aspects of the RNASCOPE-ish technology are the target probe design strategy, wherein we can design a specific target probe against any gene in any species of interest. The second aspect is the signal amplification system, wherein we have fine-tuned our amplification strategy such that we can suppress the background signal and amplify the specific signal to give you high signal-to-noise ratio. And finally, the third aspect is the detection and quantification strategy, which allows the customer to visualize RNA targets as a single molecule with single cell resolution. And this can be done on a fluorescent as well as chromogenic platform. Finally, our platform allows the flexibility to detect targets such as mRNAs, long non-coding RNAs, splice variants, highly homologous sequences, point mutations, and microRNAs. Our platform also allows the flexibility to utilize this technology in an array of sample types, which include FFPE tissues, fresh and fixed frozen tissues, cultured cells, and PBMCs. So in this next slide, I want to take some time to talk to you about the two key aspects of our technology, which is the probe design and signal amplification system. As you can see on the screen, I have depicted the RNASCOPE probe as a Z molecule, mainly to highlight the different aspects of the probe. The target binding site, as the name suggests, is designed to be complementary to the target sequence of your interest, and the preamplifier binding site is where your amplification tree will be generated. When we design our RNASCOPE probe against your mRNA targets, we design them such that they are pooled probes. We design them such that two Z probes have to bind in tandem next to each other on your target sequence. This allows for development of the binding site for your preamplifier molecule. Once the preamplifier is bound, the amplifier can bind onto the preamplifier, followed by which the chromogenic or fluorescently labeled probes can then bind on to the amplifier. Now, this allows for forming a amplification tree that allows for detection of even the lowest expressing targets. This strategy allows for really high sensitivity of detection. As I mentioned, the two Z probes have to bind in tandem onto your target sequence. Unless they bind in tandem, the preamplifier will not bind onto the Z pair. So if a Z probe not specifically binds to a sequence, the preamplifier will not be bound to the Z probe, and there won't be any amplification tree formed. As a result, no signal will be detected. Now, this ensures high degree of specificity. So combination of the probe design strategy and the amplification system ensures high signal-to-noise ratio. Our RNASCOPE technology has grown a lot in the last few years. It now encompasses three unique assays: the RNASCOPE assay, the BASESCOPE assay, and our newest launched microRNASCOPE assay. The RNASCOPE assay is mainly for targets that are over 300 nucleotides in length. We normally design the probes for the RNASCOPE assay as pooled probes of 20 ZZ pairs. The BASESCOPE assay is a specialized assay designed for detection of splice variants, point mutations, and highly homologous sequences. Because we're targeting a smaller region, we normally design a 1 to 3 ZZ pooled pair of probes for this particular assay. And finally, our newest launched microRNASCOPE assay is for really small RNA species between 70 to 50 nucleotides in length. And this includes a completely new probe design strategy and is mainly for detecting targets like siRNAs, microRNAs, and antisense oligos. In this next slide, I wanted to show you the broad product portfolio that we offer. As I mentioned, we offer the RNASCOPE, BASESCOPE, and microRNASCOPE assays. All of these are available in different formats and platforms. The RNASCOPE assay is our most flexible assay, allowing the flexing capability from single plex to up to 12 plex, which means you can visualize a single target of mRNA, or in case of fixed and fresh frozen samples, you can detect up to 12 targets on the same sample. Now, this becomes very important in cases where there is, whether your samples are precious, and in tissues such as brain, wherein it is important to conserve brain sections as you are in a different region of the brain as you're sectioning through it. Our BASESCOPE and microRNASCOPE assays are also available in different formats, including automated formats wherein our RNASCOPE and BASESCOPE assays are available in both manual and automated formats. For our RNASCOPE assay, it's available in chromogenic and fluorescent platforms as well. Currently, we have over 26,000 probes in our catalog, which means we have already pre-designed probes for various targets. But if you're interested in a probe or target that is not in our catalog, we can design it in less than 2 weeks. Moving on to the applications of this technology, because of the flexibility that we offer in the flexing capability and the sample types that we can assess, we've been extensively used in numerous applications, which include detection of non-coding RNAs, splice variants, detection of CDR3 sequences on T-cell receptors, visualization of GPCRs, which are especially known to have really poor antibodies, and interrogating mRNA of GPCRs is a great alternative to visualizing targets of interest. In addition, because it's an mRNA-based technology, we can detect the cellular source of secreted proteins by visualizing exactly which cells are producing these secreted factors. Finally, we have been extensively used as a validation tool for single-cell RNA sequencing to map these genes with tissue morphology and spatial context. And because of this, we've been widely applied in different research areas, including basic science research, for infectious disease research, for biopharma discovery and development, and in cancer and clinical research. For the purpose of today's presentation, I will be giving you a couple of examples of how this technology has been successfully used in some of the cell and gene therapy strategies. To start off, I would like to give you an example of a collaboration between ACD Biotechny and Bluebird Bio. In this collaboration, Bluebird Bio was developing CAR T cell therapy against 2 targets, BCMA and ROR1, especially for solid tumors. Using the RNASCOPE technology, they were able to design specific probes against 3' UTR of the CAR vector. This allowed them to visualize the CAR T cells in situ and track the CAR T cells in the tissue of their interest. The idea was to understand if these CAR T cells are getting delivered to the tissue of interest and whether these CAR T cells are having the desired effect. That is, whether they are having the on-target on-tumor effect or whether they're showing any kind of on-target but off-tumor effect. And it's very important to assess this at the preclinical level to understand if there's any potential toxicity from these therapies. In addition to visualizing the CAR T cells, RNASCOPE was also used to design probes against soluble factors like granzyme B and interferon gamma, which are factors released by your activated T cells, indicating engagement of CAR T cells in presence of the antigen. So in this slide, the mice were injected with the anti-BCMA CAR T cell therapy, and we are visualizing the xenograft tumor in this particular image. On your right-hand side panel, we are visualizing the BCMA antigen using the RNASCOPE specific probe. And as you can see, the BCMA is visualized as punctate dots, indicating that, as expected, the xenograft is positive for the BCMA antigen. And on the left-hand side panel, we are now detecting the CAR T cells and granzyme B. This was done using our chromogenic duplex assay, where we visualize 2 targets together. In red is the CAR T cell 3' UTR, which is indicated again as punctate dots, indicating that the CAR T cells are present in the xenograft. And the granzyme B is indicated in green, which shows that in presence of the BCMA antigen, the CAR T cells are indeed engaging and are active and are secreting granzyme B. And this is great because this is exactly what you want. You want the CAR T cells to engage in the presence of the antigen, and that is exactly what we observed. Now here, using the same tissue, we used our multiplex assay to visualize multiple targets simultaneously. This is a fluorescent-based platform, And in this particular example, we've actually combined our RNASCOPE-ish with immunofluorescence. The natural complementarity of ISH and IF allows to combine these two workflows, and a lot of publications utilize this particular workflow when they want to visualize their RNA and protein targets simultaneously. So in this particular example, we have visualized the CAR T cells in combination with granzyme P and interferon gamma, and we were also able to identify T cells by using the CD3 antibody. So what this has allowed us to do is distinguish between the endogenous T cells and the CAR T cells, which are indicated in green. And then we can also see which of these T cells are active by showing positive staining for granzyme B and interferon gamma. So overall, the BCMA CAR T cell therapy showed a success and looked very promising. But on the other hand, when we looked at the anti-ROD1 CAR T cell therapy, we saw that the researchers observed that the mice were showing some pulmonary and hepatic toxicity. And when they used ISC to understand if there was any antigen expression in the mouse lung, they were not able to detect any signal. But when they used RNASCOPE-ish to visualize if there was any basal expression of ROD1 in the lung, they were indeed able to see some degree of ROD1 expression in the lungs, which was leading to the pulmonary toxicity. And this is an example of on-target of tumor effect, which can be very serious in cases of CAR T cells. So here, in the presence of ROD1, the CAR T cells are present, as you see on the left-hand side panel, and the positive signal of granzyme B indicates that they are also engaging, causing the pulmonary toxicity. In summary of this particular study, what they observed was the anti-BCMA therapy seemed to be successful in reducing tumor burden, as you can see from this graph. On the other hand, the anti-ROD1 CAR T therapy was not as successful and showed exponential growth of tumors, of these xenograft tumors, comparable to the vehicle-treated animals. And this was in line with what we saw with RNASCOPE staining, that the ROD1 therapy showed some on-target of tumor effects. So after that, I want to talk to you about our newest launched microRNAscope assay. This is a revolutionary technology since it's very challenging to visualize small RNA targets like microRNAs, siRNAs, and ASOs. Our goal behind launching this particular product was to help researchers visualize these small species of RNA with spatial and morphological resolution. So by using microRNAscope assays, researchers can visualize ASO and siRNA-based therapies, which are showing promising preclinical results. And I want to show you a couple of examples of how this has been used already. So this is an example of where one of our customers used the new microRNAscope technology to visualize their siRNA in vivo. So they developed two siRNA therapies, siRNA1 and siRNA2, and they delivered this to their target tissue, which was the mouse liver. And using specific microRNAscope probes, they wanted to detect whether the delivery of these siRNAs was successful in the liver. So they designed a probe against the siRNA1 therapy or oligo, and as you can see here, our negative control does not show any signal. Our liver sample that had the siRNA1 therapy shows strong positive signal with siRNA1 probe, whereas the sample that had the siRNA2 delivered did not show any signal with the siRNA1 specific probe, indicating that the siRNA1 therapy delivery was successful and that the assay is very specific in picking up only the siRNA1 delivered oligo. Another example was where this particular customer was developing an antisense oligo-based therapy for muscles, and they injected this ASO therapy in different muscle tissues of the mouse. And here we are visualizing the cardiac muscle and mouse quad muscle. We have their vehicle-treated and ASO-treated tissues. As you can see, using the microRNA the specific ASO probe, we were able to see signal only in the treated sample and not in the vehicle sample. Again, indicating that this ASO delivery was successful in the targeted tissue, and the assay was able to successfully visualize these targets in the tissue. So with that, I want to just highlight how well our technology has been adopted over the last 10 years. We have grown exponentially, and a good indication of that is our publications. We have over 2,800 publications as of last month, and we've been extensively published in different research areas, which include cancer research, neuroscience, infectious disease research, and so forth. So overall, with Maggie's presentation and from a couple of examples that I showed you, I hope we've shown you that the RNASCOPE technology can be an ideal solution to detect therapeutic targets and gene expression and vector sequences in individual cells with tissue morphology, and can help address critical questions in efficacy and safety behind nucleic acid and cell-based therapies. It can be used to visualize in vivo delivery of the therapeutic gene expression and vector sequences. And using our multiplex assays, you can visualize potential immune responses mounted by these therapies as well. So with that, I want to thank you for your attention today, and please visit our cell and gene therapy webpage on acdbio.com and reach out to us for any questions. Thank you.
Thank you, Anushka. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We'd also like to ask attendees to take a moment after the webinar has ended to take our exit survey and give us your feedback. We'll now start the Q&A portion of the webinar. First question is for Maggie. How large is the SYN promoter?
The synapsin promoter. Don't remember the exact number of base pairs at the moment, but it's fairly small. It's less than 500 base pairs, I believe.
Thank you. The next question also for Maggie. What is the homology between the mouse, CDKL5, and human?
Oh, let's see. The protein homology, I believe, is fairly high. There are some exons, of course, that are more highly homologous than others. Exon 1 in particular, I think that one's over 80% similar between the two species. But I think overall the sequence homology is around 70%, but don't quote me on that.
Thank you. The next question. Do you have a control for the half-life of the mRNA in the brain? This is meant to mean, means the control mRNA.
A control for the half-life of the mRNA? I'm not sure I understand the question.
Thank you. We'll pass on that one and save it for after. The next question. How did you distinguish between the injected and endogenous mRNA?
Yeah, so that's something that we've actually had some technical difficulties with, that we've worked with ACD Bio pretty significantly, because there is a significant homology between the mRNA of the mouse and the human version. So what we do is we've generated primers that are specific to this human CDKL5, and that allows us to distinguish between the two. So even when we use these specifically generated primers in wild-type mice that have, abundant levels of CDKL5, the levels of endogenous mouse CDKL5 that the primers are picking up on is really very small. And that speaks to the power of this particular in situ hybridization abilities that ACD Bio has, is the ability to distinguish even when there's high levels of a very similar mRNA in the vicinity.
Thank you. The next question. Why did you group the males and females together for the statistical comparisons for behavioral improvement?
So when we looked at the behavior tests right after we ran the assays, we did have them separate. And what we saw was that they trended very similarly, and that was consistent with what we expected. We looked at the males when they were about 2 months after administration of the drug, and females much longer, more like 4 months. And at those two different time points, their phenotypes are very similar. And once we compared them separately and saw that they were performing the same, we feel confident in our ability to group them together and give a little bit more power to our ability to see changes between the treated and the non-treated groups once we had the treated groups of both sexes grouped together. So we did look at them separately, and because of the way that we designed the study, they performed very similarly.
Thank you. The next question, why was PBS used as a control over an empty vector?
Yeah, so we used PBS instead of an empty vector because empty vectors themselves can be toxic. Cells don't like to be administered vector that don't have anything in it, and to make sure that what we were seeing was a true result of our vector, which was, full of the DNA that we were delivering. We wanted to make sure that we were using something that was not going to be toxic in and of itself.
Thank you. The next question is for either panelist. How do you use RNASCOPE to obtain the whole brain images that were shown in the first presentation?
I can speak to that one since those were my images. So the images that we saw here were taken on our Zeiss Axio imager, which has a motorized stage. And this allows us to take large stitched images of entire brain regions or of entire brain sections. We've since acquired a scanning microscope, an Axio scanner also from Zeiss, that has the same ability, but on a little bit of a higher throughput scale to get those big, beautiful images that you're able to see right here. It's a really powerful approach to see that broad biodistribution.
Thank you. The next question is for Anushka. What is the average length of the target binding site on the probes?
So that's a great question. So for RNASCOPE specifically, as I mentioned, we design pooled probes. They're normally a pool of 20 ZZ probes, and we, on an average, span an area of 1,000 bases.
Thank you. The next question is also for you, Anushka. What spatial resolution can be achieved to study subcellular localization of miRNA?
Yeah, so that's a great question. Because our technology is fairly new, we don't have a lot of examples showing how it's showing specific subcellular expression of microRNAs. But with our RNASCOPE assay, which is our parent technology, we have seen we are able to distinguish between targets expressed in the nucleus versus targets expressed in the cytoplasm. So we do get that level of subcellular resolution.
Thank you. The next question is for Maggie. Were the synapses more mature after the treatment?
Yeah, that's a great question. So that's something that I've actually spent a lot of time trying to work on, both looking at levels of like PSD95, postsynaptic markers, and also doing Golgi staining. I'll say that because we're not hitting very many cells, it's been difficult to tease out because the number of cells that we're actually able to move that bar on is fairly low. It's something I'm actively trying to get a good, solid readout on. And if the person who's asking the question has any suggestions for ways to do that, I'd love to hear what their suggestions are. It's a difficult thing to look at such a small structure, and we're actively trying to figure out something to make that work.
Thank you. The next question is also for you, Maggie. Are you able to evaluate the degree of off-target or other overall transcriptomic effects of the AAV therapy by sequencing only the portion of the mouse brain section with a positive signal?
I'll say that we haven't done that yet. That's something that we could definitely look into in the future, but it's not something that we've been looking at in these first studies.
Thank you. Next question is also for Maggie. Have you compared the efficiency of translation of human CDKL5 in the mouse brain?
Specifically the translation, we haven't, no. We've looked a little bit at how the cells process the CDKL5 protein, the human CDKL5 protein, but we have not yet looked specifically at the efficiency of the translation of the mRNA. We felt pretty confident that we were getting robust levels based on what we were seeing with the RNASCOPE images, but it's not something that we've quantified or analyzed any further beyond that.
Thank you. The next question is for Anushka. Is RNASCOPE able to detect both transgene and vector DNA in a cell?
Yes, that is again a great question. Yes, using the duplex assay, you can visualize both the transgene expression and the vector DNA together.
Thank you. Another question for Anushka. Can the new miRNASCOPE assay be combined with IHC or IF?
Yes, absolutely. Similar to our RNASCOPE and BASESCOPE assays, the microRNASCOPE assay can also be combined with IHC or IF in the same workflow.
Thank you. The next question is for Maggie. Does the mouse model recapitulate the seizure activity that patients experience?
Yeah, so this is actually one of the limitations that we have in using this mouse model. Unfortunately, while these mice can be prone to activation or kind of overstimulating them to cause seizures, they don't actually have that baseline seizure phenotype that is so robust in characteristics of the phenotypes of the patients. So this is one of those circumstances where a mouse will always be a mouse. It's never going to totally give you what the patient experience is, and it's one of the things that we're unfortunately unable to assay with this model.
Thank you. That's all the time we have today for questions. We'd like to thank Maggie Wright from Ultragenyx Pharmaceutical and Anushka Dikshit from Advanced Cell Diagnostics. If we didn't have time to get to your question, we will try to have our panelists follow up. As a reminder, please look out for the survey after you log out to provide your feedback. If you missed any part of this webinar or would like to listen to it again, an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.